Happy Micro Monday. I'm going to run through labs 4.1 and 4.5. So we're going to do your phenol ethyl alcohol plates and your eosin methylene blue plates. And these are going to be the same setup, just streak plating, so we're going to use a technique called streak stab. And then you're going to get these in the incubator and you will read the results the following lab. So I'm just getting my loop nice and hot. You're going to be using E. coli and a new one to you called S. epidermidis. E. coli is your gram negative and the S. epi is your gram positive. The point of these two labs is to identify whether bacteria is gram positive or gram negative. The PEA plates, phenol ethyl alcohol, they promote gram positives and they inhibit gram negative. And then the EMB plates stands for eosin methylene blue. They're the opposite. They promote gram negatives and they inhibit gram positives. This is not an all or nothing. It's not a presence absence. You may see growth of both the E. coli and the S. epi on both plates. But again, the point of this media is that it inhibits one or the other. So you should see more gram positive on the PEA plate. So in this case, S. epi should grow better than E. coli on the PEA plates. And with the EMB, it's the opposite. We should see more E. coli compared to the S. epi. So this is called selective media. For each of these labs, you're going to get 2 plates. So your group will probably have 4 plates in front of you total. One's going to be labeled for the lab, and the other one's going to be a nutrient auger. So I'm going to go ahead and do the PEA plates. So they're going to be labeled PEA, and your nutrient auger will probably have an NA on there. I'm going to have you put both the gram positive and the gram negative on the same plate. So this is a little different labeling system. Basically make these little trisections, and then I'm going to go ahead and label for each bacteria. So I got E. coli, S. epi, and then the third area I'm not going to inoculate, so that's going to be our control section. And again, it's because you want to compare growth, and it's not going to be presence absence. On this plate, since it's the PEA plate, it promotes gram positive. So again, we should see more growth in the S. epi than in the E. coli, and certainly the control. I'm going to do the same thing from my nutrient auger plate. Get 3 sections, hopefully approximately the same size. And then label. Notice when I label, I'm sticking to the edge of the plate because I do not want to obscure the field of view. Okay, well labeled, and then it's just a matter of streak stabbing. So this is going to be a zigzag across the surface of the plate the way you're used to streaking. Just at the end, I'm going to do this last little touch where I dig the loop midway into the auger. So I don't want to touch the bottom of the plate, but I want to get it inside the layer of auger. I'm going to set up my PEA plate first. Actually, I'm going to leave that lid on until I'm ready to lay down the bacteria. I'm going to do E. coli just because it's first up. Open this. I'm going to run it through the hot air real quickly just to keep that air pressure gradient, preventing contamination. Okay. Loop flames. I'm going to go into, you're going to get a slant. So I'm going to touch it to the side, let it sizzle, cool down the loop for a sec. I'm going to drag this up through the agar. And I'm just going to zigzaggy back and forth. And then again, I'm going to do this last little maneuver where I stab it, just kind of puncture, and then go lateral. So down a little bit and then in. Okay. Put the lid back on my dish. While I've got my E. coli out, I'm going to go ahead and do my nutrient agar plate. And again, sizzle, 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 drag it through. That loop nice and laden with E. coli. Streak, 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 streak. Just a little bit and then stab. I'm really not streaking the whole area. These plates are only going to get 24 hours in the incubator, so I'm able to be a little more heavy-handed. All right, so E. coli's done. Back on the nutrient agar plate. So don't want it to sit exposed to the air. Run my SFB through the flame really quickly. Get that column of hot air. Get my loop. Sizzle. The same thing. Gonna go down where I see the growth. Pick up some growth, not gouging the agar. Get some SFB on that loop. SFB section, just a couple of zigzag and get it under there. Put the lid back on, flame my loop. Now I'm going to do my nutrient agar plate. Same thing, sizzle, scrape on the surface, get that bacteria. So I can put it down, nutrient agar plate, epi section, zig and a zag, and then maybe a little zig again, and then right under the surface. Come on, baby. Right in there. What that's doing is giving it an anaerobic condition. So again, we're comparing growth, just giving both an anaerobic and an aerobic condition. We don't have an anaerobic incubator, but that's a stab technique is a little cheat to create anaerobic conditions. So it'll just give us a little bit more information. It's really going to make a difference on the next lab, the EMB lab. So let me properly get my SFB closed. So this is it for the PEA lab. This will be done. You've got your nutrient agar plate, it's got E. coli, it's got SFB, it's got a control section. You've got the same thing on your PEA plate, your SFB, your E. coli, and your control section. And then you're going to put them in the incubator, top down, upside down, upside down. Okay. Next, you would do the exact same thing for your EMB plate, and I'm not going to run you through that. It's the exact same process. Zig and a zag and a little stab to get under the surface. The only difference is that your EMB plate is really pink. Right? And so this isn't going to be what yours looks like. This is for a different lab from soil. But you're going to set it up the same way with the trisections. Label it for SFB and E. coli. Give it a control. For each section, you'll lay down, transfer your bacteria, a streak, zig and a zag, and then you'll stab it, get it in the incubator, top down, and then you're done. So it's an easy setup. Next lab, we'll investigate the difference between the gram-negative and the gram-positive. And again, it's not about presence-absence. It's going to be about more or less growth. All right, see you in lab.